Good morning, afternoon, or evening, everyone, for joining us today. It is a pleasure for me to introduce and interview Stuart Peltz, the founder and CEO of PTC Therapeutics. On August 7th, 2020, the FDA approved Evrysdi, risdiplam, for the treatment of a spinal muscular dystrophy for adults and children 2 months and older. I like to quote the FDA, Evrysdi is the first drug for this disease that can be taken orally, providing an important treatment option for patients with SMA, following the approval of the first treatment for this devastating disease less than 4 years ago. And that quote is from Billy Dunn, who's the director of the Office of Neuroscience in the FDA Center for Drug Evaluation and Research. Now, this is a remarkable achievement for SMA. There's a number of options for patients of SMA, including gene therapy, antisense, and now small molecules. So the first question I'd like to ask Stu is, what does the approval mean for patients?
Yeah, well, thanks for that. And yeah, we think it really is an important therapy for patients. I mean, there's clear efficacy in these patients, not only for type 1 or infants, but as well as I think the results have shown in a broad population that includes both adults, children, and infants, that you saw improvement in all of them. So it's a broad range of patients, and this is really the first ever placebo-controlled trial for patients that can have who are adults who have these treatments. So it isn't limited only to a certain age or population. We had people were treated in our trials up to 60 years of age. So that was really, I think, really important. And we did this in part, as you know, we worked with the SMA Foundation and other groups and had very much input from patients. And so it was really, I think, the first ever clinical program that was designed and implemented with not only trying to get it approved, but with the patient and caregiver voice in mind. And the fact that it's orally bioavailable and systemic is really critical, because I think it's become very clear now that while the CNS is the first that's heavily impacted and you see the consequences of that disease, it's very clear now that other tissues and organs are affected, and that the SMN protein has wide effects, so that you see muscle, you see that muscles aren't the same, bone, bone, pancreas, many examples of there are effects in these patients beyond the CNS. And the fact that this is oral and systemic, so that the drug can get to all the organs and tissues that are affected, increase the levels, is good for patients.
That's great.
And then I think the other important is that it's in the days now of where we're in the middle of a pandemic, the fact that it is oral, could be delivered directly to the patient, where they don't have to go to the hospital, is key. Is really important. And
the second question would be, briefly, what's the science behind the drug?
Yeah, the science, I think the big message here, the importance of this drug is that it's the first ever small molecule that modulates splicing. That had never been done before. So I think the importance is that, there was this prejudice that you couldn't modulate splicing with a small molecule, because it's a process that occurs over and over again in cells, and people think that you can't modulate that, and that's just not true. And so I think the fact that I look at this, there's that book Loonshots, a big idea that most people don't think would work, but when it does, it's transformative. And I think this now opens up the whole field and looking at for small molecules that modulate splicing. And I think from our perspective, we started out as an RNA biology company with really now thousands of years of experience in this area. And so that's really, I think, that's the sort of the big idea that came out of this.
Mm. And so in the course of the discovery and the development of the drug, what were some of the challenges that you had faced?
Um, well, first of all, there's lots of challenges when you think about from the science aspect of this. Um, Clearly it's an orally bioavailable small molecule. It's in an area of biology that hasn't been tested before. So there probably weren't a lot of chemical molecules that were made that could affect the function. So we had to build up both the library and identify molecules that had some activity and then build it out over that. And then you have many of the issues that occur in high throughput screening with small molecules is you have to improve its potency, make sure it gets through the blood-brain barrier, make sure that the efficacy is strong, that it gets into all tissues. So we've sort of really brought this in the forefront, and the possibilities are now immense. And that's usually somewhere between 1 and 2 decades that it takes to get there. And so this, the second one, this took about 15 years to do. But they're cutting edge ideas, and so they take time. But so much was learned as a consequence of this.
And partnership was actually important, as you mentioned, in the development of the drug, and it went beyond the SMA Foundation, included Roche, for example. So, from a drug development perspective, can you tell me the roles they played, and maybe give an example of each?
Yeah, sure. So it's interesting. The initial collaboration started between ourselves and the SMA Foundation. And they had come to actually interesting the Duchenne muscular dystrophy meeting and took a suite, and their goal was to see if companies would be interested in working on SMA. And so we went to the suite and talked to their folks, and I said, we don't know a lot about this, but this is an RNA biology problem. It's something that we probably could do. And so over time, within the year, we had a collaboration with the SMA Foundation, and this was Lauren and Denneker, and they have a child who has SMA, so they were very driven to get this done. And so they gave us funding, and also they had an advisory board and discussions, and they were so driven, it was very helpful. And then the Roche came on around 2011. We had already identified molecules that were efficacious and worked in a mouse model, and we worked with them to pick a development candidate and then go into the clinic. And it was a unique situation there in the sense of, we did this actually initially at the time when the company was an odd time for the company where we didn't really want to partner at first, but financially we had to. And there was probably 5 or 6 people around the table, and we chose Roche. And really, they were very good about it from the point of view of we had this partnership with the SMA Foundation. We did, and we said if we're going to partner with anybody, they had to include both us and the SMA Foundation as partners, and that we weren't going to just give it to anybody, that we need to be real partners. And we set it up so that the Joint Steering Committee was each of us had a equal voice, and that included Roche. So while they steered the development in the sense of doing it, it was the Joint Steering Committee's was in control. And if we said no, it was no. Or if the SMA Foundation said no. So you had to work as a group. And I think that ultimately made it better as a consequence of that. And then, it's interesting. Roche was very much wanting to push this quite rapidly, able to move multiple compounds at a time. They did things that most companies don't do. They were synthesizing multiple compounds, manufacturing it at a higher level to be ready, so to pick one or the other.
It's a risk at the same
Yeah.
time.
So they were ambitious. Um, and I think over time, the one thing that was good is we all sort of contributed it. So there was this constant sort of tension of how to do it faster, and what's the issue with, rare diseases. You tend to know less, right? The natural history isn't often as well understood. There's less patients. You really have to move, and you're very much connected to the patient community.
So
And
you
that's
have this,
all
oh, I'm
stuff we
sorry.
were used to.
So you have this partnership, that, and, I get it's clearly impacted the pipeline, at least with respect to this drug. But what about beyond the drug? Beyond the drug, does that model scale, and does it drive efficiencies or drive outcomes in drug discovery and development?
Um, so, every company probably thinks they can do everything themselves, which is just, partnerships are always can be useful at times, especially if you lack certain expertises. We're a very different company now than we were when we started in 2005,
Mm.
right? Where we were probably 150 people then. Um, and now we're almost 1,000, right? We're commercially, we have people in 25 countries where we sell drug in 50. So we're a different company now, and therefore it changes a lot, right? In a way, we're 22 years old. We built the company based on strong science development, number of drugs that have come through. We
Mm.
don't think about having to partner for financial reasons, but to partner for good reasons
Mm.
that
Mm.
are right. So we're very different than we were in 2005 or 10, right? It's not about we don't have the resources anymore. So we would do so in a very different way.
Mm. What do you see the benefits then to the patient with this, in terms of the way you approach your problems?
So the way I think what's benefit the patients and I think what we do actually pretty well is you to me it's all about innovation, right? There's a and that a, as a company, I think if you want to stay as an innovative company, that's what you have to build yourself. Because if you think about it, like I said, you take such tremendous risk when you're a small company and you have an idea and you're trying to get there. The reason I say such risk is that from idea to approval is probably a 5% chance of success.
Mm.
When you start with an idea, it's a very low probability of actually getting it commercialized, right?
Mm.
So to do that means you're taking on tremendous risk because you believe in something, right? Um, and then what happens is you get less risk, you get more risk averse because you got more to lose. And so the problem becomes though is that what doing great things is about taking risks. And so what you have to keep is a a culture that risk is good
Mm.
and um, that we know most things could fail.
Mm.
And that's why you have a pipeline of things with a large number of products, so something will succeed. And so that's the mindset and once you lose that innovation, you're in trouble. And what you should do is be careful about quitting too soon because most of the doors you open are wrong. And so you got to get time, you got to give people time to find the right doors to go
Sure,
through.
sure,
And that often takes time.
sure. Okay, let's shift to the regulatory aspects of for risdiplam. I mean, you had the FDA gave a fast track designation, priority review, orphan drug designation, and then you were also rewarded a rare pediatric disease priority review voucher. So, in your estimation, how did those designations accelerate the drug development of the drug?
I think they're important from the point of view is they give you what you really want is interactions with the regulatory authorities so that you can both understand where they're coming from and educate them. And the more opportunity, what a lot of these do is give you more interactions as such, because of speeding things up. The more they need to talk to you more frequently, and that's important because at the end of the day, you're working in orphan diseases, which means there's usually not much treatment options. They don't know the diseases well, and you need more time to talk to them about things. And in this case, they had a couple of drugs beforehand in terms of the field, so they were a little bit more aware. So they were, because of the interactions and the discussions, was more aptly to be able to rapidly go through the process. So that is what helps. The pediatric voucher is important for other people in particular because it helps them move to pediatric disease and rare disease. Now we're a company that's our sweet spot. So we go into that all the time, right? But not everybody does, right? And so this is helping, in general, companies to go into areas that they might not otherwise go into, and it gives them incentives to do so.
Okay, great. But let's discuss bottlenecks for a moment. And you've had you got your share of experiences so far, and what do you see some of the key bottlenecks in drug development? Let's talk about first SMA, and then more broadly.
Well, SMA has shown that the bottlenecks have been able to go through. Usually, bottlenecks are about like rate limiting events. What's the thing that slows you down, right? And often it's, I always talk to people and say, what's the difference between drug discovery and say physics or chemistry? It's, the principle, the guiding principle in biology is evolution versus laws of physics or, how chemistry. So, basic laws of physics, you can sit in your bathtub and start doing the mathematics and go from A to Z and make predictions. Evolution is that it's selection, and selection means you had to be there at the time things were selected, and we weren't. So it's a very different science, right? You have to try and figure out through the haze of things. I always say, when you think about people searching for gold, the people who got rich were the people who sold the picks and axes, not necessarily the people looking for gold. Right. And that's not so far from what we do. Most things fail. But when you find it, it's so valuable because it's hard to change the course of evolution. And so the bottlenecks is really the biology and what works and what doesn't, and figuring out, is this the key path and will it change the course of the disease? And then once you know it can affect it, how do you show it? Right? Because in most rare disorders, you don't know the natural history, which means you don't know what are the best endpoints. And you have to figure all that out while in an area that hasn't had natural history, that you don't have thousands of patients to test, right? I mean, SMA Duchenne was a great example. Right. For our nonsense drug, there's 1,300 patients in the US. Right. And there's an example where half of them are non-ambulatory. So if you're going to use an ambulatory, it's now 600. Right. 20% of them are under 5. They couldn't do the test anyway. So you have these small numbers to try and get answers to. So that's always a bottleneck in rare diseases.
Right. So when you think about SMA, next generation drugs, there's all kinds of excitement now about, what to do next for SMA patients. And you look at it from two perspectives. Do we need something that's more potent, or from the small molecule side, maybe better PK attributes, or where any of these drugs or treatments are going from a distribution, or should we be looking at SMA independent processes? And then the follow-up question would be, is PTC working on any of these?
Yeah. So when I think about the disease, what's interesting is what got everybody excited in SMA is that the type 1 patients, which are, in a sense, are like floppy babies that never can lift their head and
Right.
died by 2 years of age, right? And and our drugs transform them to be able to not only lift their heads, roll over, sit, stand, walk, and you saw that dramatic change, right? So
Mm-hmm.
these drugs are effective. Now, what people will say, is that, well, you don't see as big a change in type in the older patients. Well, that's because, they're more, things happen. They're already more developed. They may have contractures, or they may have scoliosis.
Mm-hmm.
They may have lost muscle. So even though it's helping just as well, it may take a longer time to see, or sometimes you lost so much muscle, how do you improve them? So
Right.
at the end of the day, what you need is other things. That's the way I think about this.
And other things would be this SMA.
Regenerative of muscle. I mean, if I,
There would be a, I'm sorry, there would be SMN independent processes.
yeah, so let's say one of the problems, they don't have enough muscle, right?
Mm, yeah.
So, and it's tough to grow muscle as you get older.
Mm,
But if you have drugs
right.
that induce
Right.
satellite muscle cells to grow more,
Right.
and you can grow muscle at the same time you're treating them, these people would do much better. So regenerative medicine is going to be an important aspect of this,
Right.
whether it's a pill or cell. So and we're very interested in that. I'm
Okay.
interested in treating the whole disease. And so one of them is, we've now treated the underlying cause of the disease, but how do I help people who are later stage in their disease and need not just the SMN protein, but may need more muscle
Right.
or nerve development or growth, or right? Those are, to me, the next pioneering challenges.
And what kind of lessons have you learned from the SMA program? And do you think these are the lessons learned are portable to other indications as well?
Um, Sure. I think, lessons learned are, is earlier to treat is better. You have more room to improve, obviously. Therefore, that means newborn screening and patient identification at an early age. Let me give you an example. SMA type 1 are easier to find because you see them almost after birth. Duchenne patients is another disease we work on. They're not usually diagnosed till around 5 years of age. That's the
Mm,
average age of a patient. Well, you've had a lot of development by that. You get them earlier, you'll do better with them. Um, and then, from a drug development perspective, if you think about many drugs, you can help a patient by slowing or stabilizing the disease, right?
right.
But that's a lot harder to see,
Right.
right? It
Yeah.
takes longer. Your endpoints have to be better. You have to be better, right? So being able to um, being able to get better measurements that let you do faster, like SMA in some ways made it easier because you could see improvements above where they were.
So related to that is, in order to, achieve those desirable goals, what kind of key technologies would be needed in your estimation for SMA? And then, thinking beyond SMA, what kind of technologies you think are to be critically important for going after, the kinds of diseases that you're interested in?
Yeah, so we think about it one in two ways. One is the underlying cause of the disease, and then the effects like Duchenne or SMA, muscle, right? Grow more muscle. How do you get satellite cells to continue to replicate and add muscle?
Mm-hmm.
So either cell-based therapies or small molecules that induce muscle growth. Same with nerves. How do you reestablish nerve growth? Is what, a centimeter a year? Well, that's slow. How do we
Mm-hmm.
speed that up? Um, can we do it with small molecules or other growth factors, or um, and
So the underlying biology, understanding the underlying biology becomes important, and you're moving that into the clinic, getting a handle on biomarkers, for example, things of that
I
nature.
everyone loves biomarkers. Everyone loves talking about biomarkers. But there are very few and far in between biomarkers that are as good as predicting things.
Yeah.
And that makes it hard, right? Because the regulatory authorities are very conservative about that usually. And so you have to use them, but then you usually have to show clinical benefit. If you can actually be great at times to be able to define biomarkers that actually predict clinical outcomes.
Okay. We're up to our time limit, and in fact beyond, I believe. So I just wanted to actually get your closing thoughts on what the future holds in your view.
Well, I think in the future, I think a couple of things. One is what I love about a story like this is there's really a lot of heroes in the story over the years, right? It took research development teams, investigators, staffs at clinical sites, the patients who participated in their trials, the families that that had us be part of this. So they're all heroes, right? So it's really it really does take a village to get a therapy to patients, and it's really tremendous to be part of that and watch it and be part of the team. And then from our perspective, look, I'm a scientist at heart, and I love seeing innovative things. So the notion of being the first to show that splicing can be druggable, and that there's now in a way a new era in being able to tackle splicing to treat diseases, and to harness the power of small molecules to do this, lets us expand our thought process to many other diseases that we can work on. So PTC is in a place where exactly where I wanted to be.
Okay, terrific. Well, I want to thank you so much, Sue, for this opportunity to interview you and to have a discussion with you around not only risdiplam, but also general thoughts about rare diseases and drug discovery and development. Thank you very much. We really appreciate this.
And thank you, Richard. It was a fun conversation.
Okay, terrific. Thank you very much.